Dr. Alpesh Patel. Dr. Alpesh Patel is the scientific liaison for Animal Free Research UK, AFR UK, responsible for managing their scientific grant funding programs and has been working at AFR UK for nearly 10 years. Before joining AFR UK, Alpesh completed his PhD in biochemistry from King's College London, investigating the mechanism of action that causes the childhood disease staphylococcal scalded skin syndrome. Animal Free Research UK, formerly the Dr. Hadwin Trust, is a UK medical research charity that funds and promotes non-animal techniques to replace animal experiments. Established in 1970, the work undertaken by Animal Free Research UK develops reliable science whilst avoiding animal testing. Animal Free Research UK promotes the practice of non-animal research through its funding, publications, science community outreach, and the media. Funding is received in the form of summer studentships, PhD students, postdocs, and strategic grants. Projects receiving funds from AFR UK ranges from epilepsy, multiple sclerosis, meningitis, breast and skin cancer, asthma, diabetes, and drug testing to arthritis, Parkinson's, lung injury, whooping cough, vaccine testing, dentistry, heart disease, tropical illness, fetal development, brain tumours, and AIDS. In August 2017, Dr. Alpesh Patel, along with the director of Triodos Foundation and a partner of the 3RS database program at Utrecht University, launched a new website dedicated to promoting the use of serum-free media in biomedical research. The website allows scientists to identify fetal calf serum-free, FCS-free media for use in all types of cell culture and for specific cell types, and also allows scientists to exchange information on the use and applicability of all FCS-free media. Here at Queen Mary, a joint venture between the Blizzard Institute and AFR UK, the Animal Replacement Centre of Excellence, or ARC, provides a unique environment for scientists to work together with the common goal to develop, validate, and apply human-based models of disease. Professor Mike Philpott and Dr. Adrian Biddle drive the cutting-edge research. On to you, Dr. Patel. Thank you.
Thank you very much for that introduction. Normally I just start off with, hi, I'm Alpesh, and that's it. So it's nice. So yeah, so my talk's a little bit different from all the talks that have happened here, but I'm more thinking about cake now. So I might have to get through this quickly so we can have some cake or something, or tell me how to make some cake or something. But so the work that we do with Animal Free Research UK is all about ending the use of medical research, ending the use of animals in medical research. So our vision is a world where human diseases are cured faster without animal suffering. We all want to be fit and healthy. We all don't want our loved ones to suffer with disease. But at the same time, we don't want animals to be harmed whilst we think we're trying to cure these diseases. So what we're trying to do is enable scientists to create the tools, create the methods, find the cures, find the causes of disease without animal suffering. We pioneer excellence, we inspire and support scientists, and we hopefully are influencing change. And all of that put together will allow us to end the use of animals and improve medical research. And as part of this talk, I'm going to talk about who we are, what we do, why we do it, why animal experiments aren't useful, and how we're making that change. At this point, I should say there aren't any horrendous animal experiment images. We don't believe in showing those. We don't believe they advance the cause, so we don't show any of that. So please be aware that there shouldn't be anything on the slides that is of that nature. So as an organisation, what do? We fund animal-free research. It's as simple as that. We have fabulous supporters. Some of you are in the room as well, who give us money, who donate to us, And that enables us to fund scientists to do the animal-free research. We're not government-sponsored, we're not industry-sponsored. We'd love to be because they have lots of money. But at the moment, we're not sponsored in that sense. So it's fabulous people like you who understand that animals shouldn't suffer for us, who help us to fund this research. We champion replacement techniques. It's not just all about us. That we will promote and let scientists know about any research that replaces animal use. It doesn't have to just be ours. We try and influence the scientific community. It's getting easier. It's much easier now for myself to go into a scientific conference and talk to scientists about replacing animal use. It's getting much easier because the tide is changing. We educate early careers researchers. One of the big things that we're trying to do at the moment is make sure that students who are starting their careers in science start knowing that they can do animal-free research. We have been to lots and lots of universities, lots of places where the scientists are going, I didn't even know I could have a career without doing animal research. So that's what we're trying to do. Develop partnerships, raise awareness, and more importantly, fundraise. Like I say, without any money, we can't do the work that we do. And as a lot of you may know, science is expensive and getting more expensive. What are the challenges? Well, here in the UK, there are almost 4 million animals that are used in research every year, day in, day out. Yeah? Right now, every minute of every day, about 7 animals, on average, are being used. We still don't have effective treatments for any diseases. You've heard this morning about to be able to cope with a disease, to be able to manage a disease, not to treat it, not to cure it, or not to prevent it, but to manage it so that you can live with it as a lifelong disease. The drug development pipeline is at an all-time low. A few years ago, it cost lots of money, 6 or 7 digits, to produce a drug, but about 200,000 to 500,000 pounds per drug. That number has gone to over £1 billion to get a drug from a compound to the NHS where we can be prescribed it. It's just, that number has gone up and the number of drugs, the new drugs that are being produced has gone down. I haven't got the graphs. If anyone wants to see the graphs, I can show you those later. The other issue that we face is that animal testing is considered the gold standard because it's the way it's always been done. So as an industry, as a pharma company, as a university researcher, you will do animal testing, you will do animal experimentation because that's how it's always been done. Whether that's the right way of doing it, whether that's the wrong way of doing it is irrelevant. It's the way it's been done. And so that's what we're challenging. We're challenging the fact that you have to put it in a mouse. And some of it comes down to some members of the public are unaware of the problems. They just think, oh, because it's the way it's been done, it's the way it should be done. Now, the analogy that we have for that is many years ago, some people do it now, but many years ago, we all used to go around via horse and cart. It's a perfectly acceptable way of getting from A to B. Would we do that in this day and age now? We wouldn't, because we've developed new tools, better methods of transport. So we're not saying the horse and cart is wrong. What we're saying is the horse and cart is obsolete, given what we have at our disposal today. And that's what we do at Animal Free Research UK, is tell people that that's now obsolete and we need to do something better. So what the solutions we work towards? Human relevant research for human diseases. It's all about human relevancy. If we want to cure cancer in humans, if we want to treat cancer in humans, we have to look at the research that's looking at the human. There is a very famous quote by one of the directors of the NIH. The NIH is the National Institute of Health in America. It's one of the biggest research organizations in the world. One of the directors of the NIH has said in the past, we've been curing cancer in mice for decades. They can do it. They can take a mouse that has cancer. It might have human cancer because it's been given human cancer, but they can cure it with drugs, with treatments. But we can't translate that to the humans yet. So that's what we do, yeah? It's human relevancy. So the new animal replacement methods are not just about not using animals, but about saying, this is a current use of animals, this is how we can replace it. To gain support for animal free as the gold standard, that's a very a very succinct statement, but when you unpack it, animal experiments, because they've been taking place for so long, have never been validated. They've never been proven to be the right way of doing things. But with the current legislation, you have to prove that the non-animal method is 100% valid, is 100% effective before it will be adopted. And so that's what we're trying to do, and that's a regulatory thing. Develop the animal-free scientists of tomorrow and educate the public, and we've already spoken about those things. And the question we ask of everyone, of the public, of the scientists, of the industry, of the researchers, of the people that give the money is, if you couldn't use animals, if you couldn't use animal-derived biomaterials in your research, what would you do? How would you do what you're doing? Let's open that discussion, let's open that dialogue, let's open that that debate, that discussion to say, how could you do it? And that's the first step for us to get them to thinking about it. Because a lot of scientists, and apologies to the scientists in the room, and I'm not tarnishing everyone with the same brush, but a lot of scientists will say, well, that's because my professor has told me to do it that way, or that's the way it's always been done, or that's the way the research papers say I have to do it. And they never question it. They never query it. They never say, is there a better way of doing this? Is there a more human relevant way of doing it? And so that's the question we ask at Animal Free Research UK is, be innovative. Let's be at the cutting edge. We're talking about veganism, and we're talking about being on the cutting edge of that, and how it's the next wave, and how if you are a vegan, you're forward thinking, you're progressive. Well, as Animal Free Research UK, we say the same thing about the science. Let our science, the research that we fund, be innovative, be progressive, be forward thinking. So the question that some people might ask is, why should we use human models for human disease? Well, before we do that, let's take a slight step back and talk about some animal statistics numbers, just for your information. Again, one of the things we always do is make sure that people are informed. Just like all the earlier speakers, it's all about information. It's making sure you have the right knowledge so that when you speak to people, when you talk to people, you're armed with the right materials. In the UK, or in Great Britain, I should say, because this does exclude Northern Ireland, in 2017, which is the last year for which we have numbers for animal use, the 2018 statistics are actually coming out next month. 3.7 million animals were used, or 3.7 million procedures that took place. Oh, that's okay. 3.7 million procedures took place. And that's comprised of two different things. What we would call an experiment, where you actually do something to an animal, and the other half of it, or more than half of it, is breeding, is actually breeding these animals, not for use. Those 1.9 million animals you see out there on the right-hand side, they aren't then used in experiments. They're bred, they're then surplus to requirements, and so then they're killed. That's their life. And a lot of that is breeding for genetic modification, trying to make these animals more human or more realistic to humans. So more than half of all animal use in Great Britain in 2017 was breeding, was waste. We talked about food wastage. We talked about how inefficient it is to grow crops for animals to then eat the animals. It's the same thing in science. There's a huge amount of wastage that goes on. This is just a graph. The numbers on the side are millions, but this is just a graph of the animal use over time since 1995. The blue lines are the experimental procedures, the experiments that take place, and they're on their way down. They're going down slightly. What's causing these huge animal numbers is the orange bar, is the breeding, the creation of genetically modified animals. If it wasn't for this creation of genetically modified animals, the amount of animal use that's actually taking place in Great Britain would actually be on the way down. We would be making more of an impact. But because of genetic modification, unfortunately, the numbers are up there at the sort of 3.5 to 4 million. And that's just where we are at this point in time. And this is what we as Animal Free Research UK are trying to work towards reducing those numbers. Humans are not like other animals. So if we want to look at human diseases, we have to look at humans or human relevant systems. There are lots of differences. In this room, there is not one person in here that's exactly identical to someone else in this room. So if we're not identical, how are we in any way, shape or form identical to a mouse a fly, a worm, a chimpanzee, a monkey, a cat, a dog, any of those animals. Okay? Again, when we introduce diseases, so when they're studying cancer in a mouse, for example, they don't give it mouse cancer, they give it human cancer. Now, we have a research project that I'll talk about later that's looking at breast cancer, but they don't give a mouse breast cancer. They'll give it breast cancer, human breast cancer cells, and inject it under their skin, so because it's easier to look at. Yeah, so it's artificial disease. It's artificial disease progression. Poor animal experimental design. There is a lot of wastage that goes on. Unfortunately, like I say, if you're looking at certain things, sometimes it's just they're a poor experimental design. And again, there's poor human relevancy, and this is one of our big things that we work towards. Genetically modified mice are not human. Genetically modified mice are genetically modified mice. Yeah, and again, certainly we all know, those of you who have dogs, you cannot give a dog chocolate. Yeah, you cannot. It's poisonous to them. Yeah, it's bad for them. So what if there's a drug that's been tested on a mouse that kills it because it's poisonous to the mouse? But if they'd done it in a human relevant system, it actually worked. The drugs might not be getting to us because it's killing the animal, even though it may not be toxic to humans. The classic story is about the guinea pig. We always talk about guinea pigs in experiments. Yeah? If penicillin had been treated on guinea pigs, if they'd been tested on guinea pigs, we wouldn't have had penicillin because it's toxic to them. Yeah? But so it's just something to think about, and this is what we try and speak to scientists about. I've already talked about a lot of these things, so I'm not going to spend too much time on this slide. But again, one of the battles that we do have is legislation. The law requires, before the GPs and the doctors in the room can prescribe any medicines, the law requires that it has to be tested on 2 animals, one of which has to be basically a dog. It has to be an animal with a vertebrae. So it's mice and another animal. And that's just the world we live in, unfortunately. So there is some legislation as well. But again, the last line on that, 13% success rate for new drug development programs. Yeah, it's very low. Lots of times you will hear this number of 92% of all drugs that go into human clinical trials fail. And these sorts of things. But there's a 13% success rate for new drug development. And that is unsustainable. We talk about unsustainability when we're eating. The drug delivery pipeline is unsustainable. And there are a lot of reasons for that, but one of them is the reliance on animal models. So how can we achieve that replacement? We need to fund it. We need to develop the tools. It's very hard. If I spoke to any of you and said, stop doing what you're doing, whatever that is, whatever work you're in, if I say to you, stop doing what you're doing because it's wrong, the very first question you're going to ask me is, okay, well, what do I do instead then? And this is what we do. We give them the alternative. We give them the replacement. We fund that research. Don't use the mouse because we now have this new technology that can do that instead. So we fund that research. In terms of the actual research, to engage them, to be innovative, to phase it out slowly. This isn't going to happen overnight. I would love to not stand here in 10 years, 20 years, 30 years' time because I don't have to be, because I'm out of a job, because I'm stacking shelves at Tesco's, because no one uses animals anymore. That's my goal. Yeah, is to be out of a job. It's very strange, but my goal at work is to be out of a job. It's brilliant.
You can come and make cake with me, Alpesh.
Oh, fair enough. There we go. All right, I'm done. But it's a phasing out. We have to be pragmatic about it. So it's phasing out the animal research. It's that cycle you can see on that side. Give us the money to do the research, change people's minds, influence the public, gets us more money, gets us more research, helps us change policies quicker, helps more people understand who we are and what we do and why we do it, and it goes round and round, and it's self-fulfilling. And that's what we do. You may be asking, how can we replace animal experiments? This is a very busy slide. I will break this down over the next few slides. There are lots of ways we can replace animal experiments. It is not a one-for-one, like-for-like replacement. We don't take a mouse and replace it with something else. We don't take a particular experiment and replace it with a direct thing. There are lots of different ways you can intermix the two. It gets very messy, but as scientists, we can discuss it and we can tease it out and we can work it out. Anything from clinical trials to 2D and 3D modeling, microorganisms, maths. I hate maths, but you can do maths. Imaging techniques, so actually taking images of the body, population studies, genomics, all the omics that you might have heard of, genomics, metabolomics, proteomics, biomics, and all the other omics you can do. And the new buzzword, organs on a chip. And I will talk about all of those. What we also talk about is in vitro techniques. In vitro techniques are just experiments that take place outside of the body, in a lab, on a petri dish, on a tray. And the reason this picture's up here is a lot of people think that we're trying to take the ladybird and replace it with something else that looks like a ladybird. Yeah, and it's not what we're trying to do. Yeah, we're not actually trying to do it. We're not trying to take an animal study and replace it with something that looks like the animal study. We're trying to take an animal study that's flawed, fundamentally flawed, and replace it with something that is human relevant, that will give you the answer, the data, the answer to your question about a human condition. Yeah, and there are lots of ways of doing it, and these are just some of them. So what I'm going to do now is just talk about some of these methods, yeah, how they work. I'm not going to go into huge detail, but I'm going to talk about the projects that we fund that work in these areas, just to give you a flavour of how we do the research, or how we fund the research that we do, and how scientists across the UK are trying to replace animals. So the first one is imaging and scanning technologies, and these are things that we've all heard of: MRI, CT scans. You may have not heard of one called MEG or ECOG or TMS. TMS is brilliant. TMS is you zap the brain with magnetic waves or electrical waves and it turns bits off. It's brilliant. So you can use all of those techniques, all of those diagnostic techniques that you might go to the hospital for. Yeah, you might even X-rays. We haven't put X-rays on there, but you can use all of those techniques to image the brain. We funded research over the last few years looking into general neuroscience. How does the brain work? But looking at migraines, one of our researchers found that there's a very specific brain signal, a very specific brain pattern that occurs just before an episode of a migraine. Yeah? And so they're now teasing that out, and can we interfere with this little signal that happens in human brains to stop people getting migraines and stop that progression of migraines? It's been used in pain relief studies, epilepsy studies, even kidney disease. There is now a protocol that's been developed. So you can do an MRI scan of a patient's kidneys and just based on that, do a whole load of research on that, but also do lots of diagnostic tests just now on one scan. So rather than having to go through multiple times or do repeat doses, and your doctor turns around and says, go for a scan, and then go, all right, you'll have to take one another 3 months, and you've got to wait 3 months for another scan. You can now do it in one scan and get all the data you need for kidney disease. MRI, some of you may have been for an MRI scan. It's very strange to be in an MRI machine. But what we've used MRI for is looking at chronic kidney disease. We all know if you've got chronic kidney disease, 1 in 8 adults in the UK actually suffers from some sort of chronic kidney disease. So we funded a project at the University of Nottingham, and this is the protocol that I'm talking about. It's a multi-factor kidney MRI protocol, and it provides you all the information that a surgeon or a doctor or anyone who's treating you will give you all the information about your kidney structure, its function, the blood flow, so they can tell exactly at a glance what's going on with that. There are lots of pros because it's directly relevant to the patient. You can image the human's patients. You don't have to look at an animal's, a mouse kidney to look for this. In disease, the scientists or the doctors can get lots more information about the kidney disease that's going on. So it's all directly relevant. And without the funding that we did, they would still be going and doing those chronic kidney disease experiments on large mammals to see how chronic kidney disease develops. The cons to this are these machines are expensive. This one that you can see here is based at University of Nottingham. That's £10 million, that machine is. But now they have it, it can be used all the time. It can be used all the time. So it's very, very useful. So MRI is very expensive, but not everything is. Computer modeling. Computer modeling is just basically sitting at a computer, not playing games, but taking huge amounts of data. The data's already out there. There's so much data out there already, and it's just churning through it and finding that needle in a haystack. Yeah, so it responds well with big data sets. We all heard about the Human Genome Project, yeah, where they sequence the genome of the human. But like I say, every human in this room has a different genome. So now the project is called the 1,000 Genome Project, where they're now trying to take 1,000 human genomes and see what the differences are. Well, you can't do that by hand. Yeah? Even Einstein couldn't do that by hand. You need computers. You need to have the tools. But once we have that data, then we have clever scientists who can create the tools that allow you to analyze that data. Then you can go in and look at human data and say, okay, looking at the patient data and looking at the background history and all the data that comes with the genome, why is this person responding to drugs? Why is that person not responding to drugs? Why is this population, this group of people have a higher risk of cancer than that group? And you can do all of those things on a computer with human data, not with animal data. You can do toxicity testing because we know what chemicals are, we know how they react, we know what they do. So instead of having to inject that chemical into a mouse to see what happens, you can actually just look at its structure and you can do those computer simulations of that. Lots and lots of other things. The biggest issue with that is it's only as good as the data that you put in. So if you put in bad animal data, you will get bad animal data out. If you put in good human data, you will get good human data out of that. And we have funded a lot of work in this area to try and develop that. So there's a big project going on in America at the moment using machine learning, artificial intelligence, call it what you want. It's computer models. I like to keep it simple. Let's not give them all the buzzwords. But they're using machine learning to predict toxicity of all of the chemicals that are in the environment. When I mean environment, I mean in our food, in our water, in our clothes, in our drugs, in everything that we do. So all the chemicals that we currently are exposed to all the time. A lot of them, and you'll be shocked to know, a lot of them haven't been safety tested. A lot of them haven't. They think they're okay because we're using them and not a lot is happening, but a lot of it hasn't been safety tested to the degree that is required in this day and age. So what governments have said is we need to test this data now. We need to test these chemicals to see if they're safe for human use. And so the scientists went, okay, that means we're going to have to do lots and lots of animal experiments. But instead of that, what the American government actually did is they actually gave them a lot of money to develop the computer models instead. So that was one of the things the American researchers have done well. Only one of them. So there is a lot of impact on that. It has this machine learning software that they're using will and has and does replace thousands and thousands of animal experiments each year. The animal experiments that are being replaced, you can see on the screen. You drop chemicals into rabbits' eyes, you feed them to rats and see what happens. Or the LD50 test, for those that don't know what the LD50 test is, LD stands for lethal dose 50. It's the dose you give to an animal, a human relevant dose you give to an animal, so that half of the animals you test on die. That's the test. So you take, for example, 10 rats, you inject them with various different concentrations, and it's the dose at which half of those animals will die, is the LD50 test. And it is a standard test that takes place. It has been replaced though. Lots of times you don't need to do it because you have other chemical data. That means you don't have to do that anymore. So, talking about some of the projects that are happening here, right now, not quite on this campus, but just down the road at the Whitechapel campus as well. So, sorry, just to say, we have funded, in our nearly 50-year history of funding research, we've funded over 210, near enough 250 projects now. This is a slightly old slide now, I should get that updated. Nearly 250 research projects of various descriptions. These are some of our projects that we've got on. We've got a brain tumour project at Portsmouth, we've got a breast cancer project at Aberdeen, medical cadavers project at Dundee, chronic pain research at Belfast, the ARC here at Queen Mary, and a diabetes project at Exeter. And I will just very quickly go through all of those and the technologies that are being developed as part of those. So our project at Portsmouth, the brain tumours, with Professor Geoffrey Pilkington. Professor Pilkington is a fabulous bloke. He's a brilliant, brilliant guy. He has spent the last 30 years of his career, his whole scientific career, not doing animal research. He's a professor, so he is living, breathing example of the fact that you can have a scientific career without doing any animal experiments. What are they doing there? Why are they looking at brain tumours? They're looking at brain tumours to do with cell culture. It's all about cell culture, this project is. Growing up cells from humans to replicate the human condition. So what cell culture is very fancy, and there's lots of associated terms with it. But in essence, it's just growing cells outside of the body, not necessarily human cells. So when someone says to you cell culture, don't necessarily assume it's human cell culture. Any cells from any animal that can be grown in a petri dish in the lab is cell culture. And there are lots of things to go with it, but one of the key things is it's actually quite difficult to maintain and grow human cells in a lab. You need to be quite specialist at what you do to be able to maintain them very well. Human cells, and again, this is one of the reasons why people haven't traditionally used them in the past. Human cells are very hard to keep happy outside of a human body. Yeah, animal cells are much happier outside of an animal cell, outside of an animal body, but human cells are very hard. The new key buzzword, there are two new key buzzwords in cell culture, 3D organoids and organ on a chip. It's just fancy cell culture, people. Yeah? It's taking lots and lots of different cell types, growing them up in a plastic dish, in a petri dish, so that they resemble what it would look like in a body. Now, people will talk about brain on a dish. They'll talk about lung on a dish. They'll talk about kidney on a dish, liver on a dish. They'll talk all these. We're not talking brain as in the whole brain. You break the brain down into its smallest component, the smallest bit that could be identified as a brain, and that's brain on a dish. When we talk about lung on a dish, we're not talking about the whole lung. We're talking about one alveoli, one little air sac, and there are millions and billions of them in your lungs. One little air sac on a dish is an organ on a chip. But you can use them because they're human, because they react just like they would in your body. You can use 3D organoids and organ on a chip to test drugs, to see how they affect them, to see what changes, to see what's changed in a diseased patient versus a non-diseased patient. And they're very, very useful. And like I say, there are lots and lots of organoids that can be used. So how are our Portsmouth researchers using 3D organoids and organ on a chip to look at brain tumours? They're actually created the world's first all-human 3D blood-brain barrier model. Now, the blood-brain barrier is annoying in humans. Its function is to stop anything nasty getting into the brain. It's great for when it's in your brain, when it's surrounding your brain, it's great because it stops all nasties getting in. It's what keeps your brain healthy. But when you have a tumour, when you have a brain tumour, the blood-brain barrier stops those drugs getting into the brain to be treated because the blood-brain barrier sees it as something foreign, something nasty, something not great. So what's our best chance at the moment of doing this? You actually have to hammer that blood-brain barrier you have to overload it with drugs so that some of it leaks through. And that's why treating brain tumours is so difficult. This is why there are a lot of side effects to treating brain tumours, because it's so hard to get the drugs across. You have to hammer the whole body, because you have to inject it into the blood so it goes everywhere. You have to hammer that blood-brain barrier so that some of it gets through. So what has Professor Pilkington done? So they've created, in a dish, in the lab, this model of this blood-brain barrier. So now they can test new drugs, current drugs, new delivery methods to see if anything is better than the current available system to get drugs across. So rather than doing that experiment in animals, you can do it in the dish, and you can do it multiple times on a table. You can just set them up one in series, one after another. Yeah. So what they're currently testing is nano delivery. So using nanoparticles to see if that's something better at getting drugs across, and they're having some quite good successes with some of these new delivery methods to get existing drugs into the brain to treat brain tumours. So what's been its impact? They've saved thousands of mice from those experiments. It's sped up development. You don't have to grow up a mouse, treat it, dissect it, take its brain out, look at what's going on, do that multiple times. You can just do this in the lab, and you can do it many, many times, one after another. So it's also, it's a very simple way of repurposing drugs. So taking existing drugs, see if they work. If they don't work, you don't go to the animal experiments. You don't need to try it on an animal experiment. And they're also using it as, they're starting to use it as a way to stop breast, lung, and kidney cancer spreading to the brain. One of the big problems is when you have a cancer somewhere else, many times that cancer will spread to the brain. And then it's very hard to treat it. So they're also using their model to see how does cancer get through the blood-brain barrier that we have and become a brain tumour. So our chronic pain research at Belfast, these are our lovely researchers, Dr. Al-Karim, Dr. Lundy, and the PhD student in the middle, Haley, are working on chronic pain research. And lots of people suffer from chronic pain. According to the numbers, 28 million adults suffer from chronic pain in the UK. And what our scientists are trying to work out is what causes chronic pain to start. Yeah? Can we diagnose chronic pain before it becomes chronic? What are the triggers that cause pain to become chronic pain? And what they found is they're actually using discard. They're using waste human tissue to do this research. So when you go to the dentist to get your wisdom teeth out, they can actually use the dental pulp that's in the middle of that wisdom tooth to do this research. So it's waste human product. It's product that we would throw away, or that the dentist would throw away. But they're using it to extract the nerve cells from the middle of our teeth. Lots of us have sensitive teeth. It's the same nerves. And they can isolate those nerves from your wisdom teeth and do the research on there, rather than doing animal experiments. And I don't need to describe the animal experiments to induce chronic pain. Yeah, that's all I need to say. Yes, and I'm not going to talk about that. Yeah. So why do we need to actually use wisdom teeth, and why can we not just extract nerves from humans to test on? We're not actually allowed. Strangely, you're allowed to experiment on animals, but you're not allowed to experiment on humans. Yeah, there are no ethical, there are no legal ways of extracting nerve cells from humans. And nerve cells across different species are different. Nerve cells in different animals are different. So by using this waste tissue, they found a completely different way of getting human relevant material to do their research. And again, what they're doing is they're doing cell culture. They're growing these cells up. Our breast cancer project at the University of Aberdeen, again, is another cell culture project. And it's slightly different. It's cell culture, but it's also looking at demographics. Although demographic studies aren't really a replacement technique, it's still using demographics. And demographic studies are studies of populations, cultural evolutions, effects like we've talked about earlier, the effects of lifestyle, diet, occupation, all of these things. Yeah, and so this is what they're doing. They're doing cell culture and demographic studies. And what are they doing there? They're looking at the environmental factors that are leading to an increase in breast cancer numbers. The average rate of breast cancer in the population is going up. And the thought is that's because of lots of environmental chemicals that are in the environment that are causing these breast cancer rates to go up. So what they're doing is they're looking at these demographic studies, they're looking at these population studies, and they're trying to isolate what chemicals are in the environment that are causing certain people to get higher rates of breast cancer, and how breast cancer tissue density is affecting breast cancer rates. So it's a very, very unique project looking at demographic studies, looking at the environmental cues, and doing some cell culture to see how that's affecting breast cancer. And again, traditional experiments would use mice. You would inject mice with human breast cancer tissue and see how that develops over time and see what affects that. So again, I've talked about most of this. So it's in a typical experiment, in a typical series of experiments, there'll be about 4,000 mice that will be used for some of these studies. And that's what we're replacing. And we're doing it in a human relevant way. And we're trying to going by what all the earlier speakers spoke about, it's about understanding the causes and preventing breast cancer from occurring, rather than just treating it and managing it for the rest of someone's life. Now, here at Queen Mary, we actually have a very unique centre. It's the Animal Replacement Centre of Excellence here at Queen Mary. It's based over at the Blizzard Institute at Whitechapel. And rather than just funding an individual project, rather than just funding a couple of researchers here and a couple of researchers there, what we've done is we've actually invested in when we call it a centre, it's some lab space in the Blizzard Institute, led by Professor Mike Philpott, second from the left there, along with Dr. Adrian Bidler, our lecturer, and two postdoctoral researchers, Mohammed Aman and Gihad Youssef. And what they're doing is they're investigating cancer. And they're looking at various different cancer research, and they're creating human-based models of cancer research. They're looking at how cancer spreads, so they're looking at metastasis to see how it spreads and invades other organs. And they're also looking at those signals, those triggers that cause cells to become cancerous. The signaling pathways in every human that causes those cells to become cancerous. They have a very, very, very good relationship with the hospitals and surgeons over there. So they get human cancerous tissue direct from patients with all the ethical treatment, ethical consideration and ethical paperwork. They can use direct human cancer tissue to look at what's going on in those cells. They also have, because you need to have the control, you need to have the normal and the disease. They also look at skin that's donated from cosmetic surgery. So the people that are coming in for surgical procedures, some sort of cosmetic surgery, they can take that skin and they can do their research on that as well. So that's how they're doing their cell culture there. Why? We've already talked about the 4 million animals. We've already talked about the fact that we can apparently cure cancer in mice, but we can't cure it in humans because the drugs don't work. The drugs don't work. And mice react differently to cancer than humans do. The way they, I would say, clear out, the way that the body responds to cancer in mice, it's different to the way humans respond to the cancers. And so looking at those treatments, looking at those ways to not cure cancer and taking place involve different pathways. And so by using these human donated tissue samples, we're saving thousands of animals from experiments where, as you can see on the screen, they will have, mice will be injected with chemicals to cause cancer. If we're looking at skin cancer, they will be subjected to sunburn through radiation, so they'll just shine a UV light on them until it causes them cancer. Like I say, I don't need to put the pictures in your head of how they look at skin cancer. Our diabetes project at the University of Exeter. It's an organ on a chip project. They don't like to call it that, we don't like to call it that, but it's the way that good marketing and PR happens. The original phraseology came from the Wyss Institute at Harvard. Here at Queen Mary, again, there is an organ on a chip network that's being led by Professor Hazel Screen here at Queen Mary to help the UK develop and promote and work on organ on a chip technologies. Organ on a chip is what you can see on the right hand side there. It's just a little plastic chip with vessels that mimic blood vessels, and on either side you can grow cells, you can grow these 3D organoids to make these organs that function very similarly to the way humans do. And again, the caveat to this is if you put animal cells in there, it will act like an animal cell. But strangely, this development has all been done with human cells. The focus is on developing human organ on a chip. Again, the reason industry wants to do it is they want to do it for personalized medicine. They could take your cells from your body, grow it up in a chip, see how you respond to drugs, give you that personalized medicine. So we talked about that. So diabetes, why is this organ on a chip project working on diabetes? About 3.3 million people are diagnosed with this, and it's currently increasing. There is a huge burden on the NHS. We've already talked about this treatment versus just managing it, just living with a disease. It's a huge cost to the NHS, and unfortunately there isn't a cure. We know lifestyle affects diabetes. You have a healthy life, you can cure diabetes, or you can treat diabetes, you do not have diabetes. But why? What's actually happening in the human body that's causing that change? It's great that you change your lifestyle and you don't have diabetes, that's brilliant. But why? What's causing that? And that's what this project is looking at. We did an initial project that finished in October 2018 that actually it changed the paradigm with diabetes. what lots of people thought in diabetes was that the cells that produce insulin die, and that's why you stop producing insulin. The numbers get lower and lower and lower, so you get less and less insulin, and therefore that's why you need to inject yourself with insulin in diabetes. What our researchers have found in this human system is the cells don't actually die, they change. They become something else. And that something else, this other type of cell, doesn't produce insulin. And this is what the lifestyle choice is actually doing. A high cholesterol, high sugar environment causes these cells to switch off insulin production, to become something else. And so you change your lifestyle, you reduce all those triggers, and the cells switch back again and start producing insulin. And that's the cure, that's the treatment, that's what's happening. So what this project is doing is, at a molecular level, is what is that switch? What is that trigger that's causing the cells to stop producing insulin and become something else? We wouldn't have found that in mice, because the cells in mice, they do change. Some of them die, some of them change, but they change to something else. And that change is mainly irreversible. It's very hard to change it back. Yeah, I'm not going to start talking about alpha, beta, and delta cells, but it's all about alpha, beta, and delta cells. Yeah, but if you want to know, come and talk to me. But it's the change in cell types. Yeah, but what this project is doing is it's looking at what's the switch. Lifestyle is great, but we all know lots of people won't change their lifestyles. Yeah, it's very hard to convince people who are on high fats, high sugars, high salt diets to change. Some of them do, lots of them do, but some of them don't. So let's understand it at a molecular level. Once we can understand it at a molecular level, technically you don't need to change your diet. You can pop a pill, and that'd be great, especially for someone like me who loves cake. Yeah, but this is the potential impact of this project. It will transform the way diabetes is treated. It's not just about eat healthily. It's not just about take insulin. It's not just about take metformin to regulate levels. It should remove the use of insulin, because if we can find a drug that stops the switch, that stops the insulin production, then insulin will be produced. We will then have cured, in inverted commas, diabetes. We won't have cured because they won't have got it in the first place. Yeah? But again, the impact on the animal use is 100,000 animals worldwide. That's an estimate. We don't know the actual numbers, probably higher than that. But what we know from looking at the research is that about 100,000 animals yearly are used in diabetes research. And that's what we're trying to work, not against, but to reduce that use. This is my favorite project. This is the project that I love. The Medical Cadavers Project at the University of Dundee. I apologise, it's after lunch, we've all eaten. There aren't any gruesome images. There aren't pictures of dead bodies. But this is brilliant. How can you do human relevant research when you are not allowed to get the ethical permission or the legal permission to experiment on humans when they're alive? You can work on human bodies that are donated to medical science after people have died. Now, the problem with that is embalming. When you embalm a body so that it doesn't start rotting, everything goes rigid, everything goes hard. It's human relevant because it's a human body, but it's not human relevant because it's not floppy. What they do here at Dundee is they're using this new method of embalming that, apologies, but it keeps everything soft and squidgy. Yeah, for up to 18 months. They can embalm these, and the reason why they say 18 months is after 18 months they actually stop using the body and they actually give it back to the family. They could actually keep it longer. They can actually keep these. And what they're doing is they're looking at medical devices. And I'll be the first to put my hand up and say I was ignorant of this fact until this project came along. I did not appreciate this, and apologies to the doctors and surgeons in the room, I did not know this. But if, as a surgeon, you wanted to learn a new surgical procedure to do some sort of stent delivery or something like that, how do you do that? How do you practice on that? If you practice on a normal cadaver, it's hard. You can't do blood flow. You can't have arteries expanding and things like that. So what they do is they fly over to Europe and they practice on pigs. I had no idea. I was like, I don't want my surgeon to work on pigs. But they do. They go off to Europe and they practice on pigs because it's roughly the same size as a human heart. So what they can do here is they have these cadavers that are, again, and I apologize, they are soft and squidgy. You can actually pump blood through them, or blood substitute. And the arteries will flex. The veins will flex. You can actually have a real-life stent inserted, and the surgeons can go in and see if their technique or the new procedure will work on a human. If there is an industry out there that's producing a new different type of stent for it to be approved to be used on humans, they can put it in these cadavers and actually see if this stent is better or worse than currently existing technologies, reducing that need for going to other countries to do animal experiments. Now, I don't know about you, but I would much rather my surgeon practice on a human before they're going to come to me. God forbid I need to go for surgery, but if I do, I'd much rather want my surgeon to have practiced on a human before they start on me for the first time. Yeah? But it's unbelievable, this project is, in terms of what they can do. I can't show you the pictures because we're not allowed to, but you can see the veins, and remember, these are dead bodies and they've been dead for a long time. The arteries and the veins will flex. So when you add a stent in there, you can see if it flexes in line with the artery and vein, so that it won't become loose. It will be a long-term solution. And again, when they're testing on these pigs don't have diseased arteries. We're not all perfectly 100% healthy. We all have underlying issues. There's always a little bit, I think, apart from a few people I've spoken to who are super fit and super great. We all have a little bit of cholesterol. Yeah, we all have a little bit of furring of our arteries, but the pigs that they test on are clean, are sterile. They're as healthy as they can be because it's easy to test on. And so they can do this. So they're accurately testing new medical devices for use in humans. And again, they're not branching out because they've been working on this for a while, but they're now working on devices to help with knees and knee replacement and all the joints in your knees and things like that. And I'm not a medic in any sort of way. I'm a scientist and I don't know anything about the knee, but there is no other model that can simulate the blood vessels that are in the knee. And any sort of injuries, sports injuries and things like that need to be treated, they can do that on this. So I've already talked about this. So like I say, the impact is you can do these procedures over and over again. You can practice over and over again. So your surgeons and you can be confident that the surgeons have done things in humans and done it multiple times, and that the devices that they're testing are fit for human use. And again, the last one on there, it saves the lives of over 30 animals per device tested. And again, about 4,000 animals per year worldwide are being used to test these medical devices. So going back to this slide again, there is a lot of ways, and I appreciate I've been very, very brief about a lot of them. There is a lot of ways that we can replace animal use. There are a lot of techniques, there are a lot of technologies out there that can replace animal use. It just requires people to ask the right questions, to have that innovative thinking, to say, if I can't do this in an animal, is there something out there that I can use? If there isn't, is there someone who will give me money to develop those tools to replace the animal use? We are on a journey. We are not at the beginning, we're not at the middle, we're not at the end, we're somewhere in this journey. But again, like I say, my aim, my goal is to be out of a job, to be stacking shelves at Tesco's. I would love it. I'd be the happiest man on the planet, possibly. So thank you very much for your time. We'll take questions at the end of the Q&A. But if anyone wants to know more about who we are, what we do, feel free to contact me. That's my email. Both of those emails come to me. Follow us on Twitter. We have a very active Twitter following. There's lots of interesting things that we tweet out, lots of interesting novel techniques and technologies that are being developed. So please do follow us on Twitter as well. Thank you very much.